वो इतना बोला वो है
रिमोट का ठीक है आधा तो रिमोट है
कोई और वैसे नहीं समझ
ठीक है वही पेरेंट का आशीर्वाद के दो ये दूसरा रूम लेता हूँ मॉन्ट क्या मॉन्टेरो का
मुझे क्लास में भी है ना इतने बाद में बाहर चलते हैं ना काम कर रहे हैं आ बैठ जाओ बेटा है ना वो पे जगह पे थोड़ा सा डिस्टर्बेंस हो जाएगा ना वो फालतू उसका काम चल रहा है ना क्लासेस में डिस्टर्ब ठीक है ना चलो बाहर
तो आपको ऑनलाइन ही है ना कहीं से चलो
हाँ अरे हाँ
बेस्ट है ऑनलाइन तो तुम आपको काम के साथ को डिस्टर्ब नहीं करना
नहीं क्लास के टाइम में
कैसा सही लगेगा गुरुजी बैकग्राउंड में मस्ती वाला करो
ये सब कनेक्टेड है चेतन
Hello all. So today we will be looking at introduction to proteogenomics live session, where we'll be discussing about the different topics that have been given in the lecture about the introduction to about in this week 9, where there have been tried to integrate about the genomics technologies and the proteomics uh, thing. So till now we had looked at different softwares and different techniques to analyze the genomics data as an individual and the proteomics data as an individual, but how to integrate. So in the different lectures of the introduction to proteogenomics week 9, we have seen that why these two are important and how these two can help in analyzing and assessing different types of data and understand the different clinical outcomes has been given. So here we will look at another aspect of which is very much currently in use, or which is thought to be of very much importance in the coming future, is the personalized medicine. So we'll be mostly focusing on how personalized medicine or personalized therapeutics, what are there, how proteogenomics is helping to achieve that goal, where personalized medicine or personalized therapeutics or precision medicine, or how these integrated technologies can lead to achieve that goal. So the what is a personalized medicine? So that now if we look at the treatment modalities, current medicine remains with that. For example, here in this paper uh, has been given, so you can just go and search this blog for better understanding of the concept of the precision medicine. So if we look at, for example, they have taken a cancer patient with colon cancer. Now one single therapy is given to all the patients. Now what happens is that these patients, effect of these drugs happens in three different ways. So the patients can be uh, subdivided once the patient is administered with this particular therapy. So the patient can be divided into three types based on the outcomes of the drug or outcomes of the treatment. One is that the drug affected the patient very well, and the cancer disease, it worked well against the disease. It helped in curing or the management of the diseases. But one thing can be that the management of the diseases, but another can be is that there is no effect. So whatever drug you are giving, the therapy has no effect on patients and it is going on. Another can be an adverse effect, where rather than curing the disease or curing the patient, it has led to adverse effects where the patient's health had deteriorated or many side effects could be seen. So the now ideal case is should be this graph, where the effect of the drug should be high and the patient should get cured, or the patient's health should get in a better way. But we cannot restrict ourselves and with the current forms of the current modalities of treatment, and it helps in getting a no effect or an adverse effect. So now the future medicine is like a kind of a mode of a personalized diagnostic. We are like cancer patients with different colon cancer. So we know that there are different biofluids. You can have different biopsies, different tissue specimens. As you will be getting the biopsies result. So you will have different tissue specimens. So you can take out the blood, you can take out the tissue specimens, you can take out the DNA, you can have the protein, you can have the RNA, and then you try to profile those patients, and then you try to target each medicine for each particular group of patients. So now the effect should be, since you are not giving or not prescribing the single therapeutic measure or single therapy, a single drug for all the patients, and all the patients, now that those three groups which show deteriorated effect or a no effect, and now given similar different kinds of drug, So now every patient will have its effect, or the ideal graph is achievable, or the ideal graph is achieved by each and every patient, and the effect of these drugs on those patients are huge, and the patients get hugely benefited. So this is kind of a more kind of a personalized diagnostics, which is very much needed, and which is think of as a future medicine, which where every person will get a kind of a personalized thing, personalized cure or treatment by analyzing their data, so that actually the germline variants or the somatic variants, or what are the variants, or what are the particular cause of the disease in that particular patient, and accordingly it will, the drugs targeting those pathways or those will be given to those patients, so that they can be administered in that way. So now this, for example, one of the major occurring diseases are cardiovascular diseases, and so what is the scope of this precision medicine and personalized medicine in the cardiovascular diseases? So here you can see that if you go through the paper, so if the precision medicine aims to offer the right treatment to the right patient at the right time. So this is the main thing. So that the precision medicine, the term is refers to that, so that the right treatment is given to the right patient and at the right time. So in cardiovascular medicine, the potential of precision medicine to applies all stages of the development, and includes risk prediction, preventive measures, and targeted therapeutic approaches. So not only the given after the disease, but also to predict the risk, and also to provide the preventive measures. So how it is helping us, so how the concept of precision medicine is gaining popularity or is getting benefited, is that with the era of these genomics and other omics that have helped us in establishing the biomarkers of the different mutations which are responsible for this. So in from there we can analyze those data and can try to identify that what type of treatment to be given for the right time. So other disciplines, not only cardiovascular diseases, but oncology and immunology have have already moved to molecular diagnostics, then led themselves to precision medicine approaches. So for like cancer and immunology, there are many. Uh, it has been to try to identify the different genomic markers, mutations, and all, and targeting those mutations are all in place. So those can be applied in for cardiovascular diseases as well. So that the different conditions like angina or other can be used. So basically the major goal to understand here is that the precision and the personal medicine can be applied to all those diseases, but focusing at the molecular level and at the symptoms. So understanding the molecular mechanisms of this particular person, that will help in administering the drugs in a better way. So this is was one of the study from integrated personal omics profiling from Stanford Medicine, which tried to identify for the personal omics profiling of a particular person, and try to identify that how that person can be administered. So for a type 2 diabetes mellitus, so which is a scientific significant health problem, and it's been one of the major causes of all forms of diseases all around the world and deaths. So in that Stanford, they had tried to identify that how the gut microbiome between the person of diabetics and healthy individuals, and direct alteration of the microbiome has shown to lower glucose levels. So when diabetics persons or the mice or persons have a different kinds of gut microbiome, but when the gut microbiome have been changed, they find out that their glucose levels become low. So so to better understand these biological changes that occur T2 to disease acquisition, they performed a plan to detailed analysis of the biological processes that occurs at a patient at risk for T2 and their microbiomes by longitudinal profiling of patients during healthy periods and periods of stress. So whenever the patients were in healthy condition and during the periods of stress, they try to find out the biological processes that occur. So differences in this gut microbiome have been noted between diabetics and healthy vegetarians, and direct alterations they could find out that they have lowered the glucose levels. So now, so the study how they designed was to establish of 100 individuals which are having a risk for diabetes, and during infected and other stressors, and less frequently during healthy periods, with a time point of 27 time points available to subjects to check on that how their gut microbiomes are getting altered from time to time, and for to analyze how will they will understand that their gut microbiome is changing or not, or at the different levels of stress or at the different levels of conditions how their gut microbiome is. So that to perform the proteomics analysis, omics analysis of the longitudinal patient samples and samples of their microbiomes. So microbiome, genetic, transcriptome and proteome content were determined in different samples for viral and indigenous microbiome populations, and then the patient genomic, transcriptomic and proteomic data were analyzed, and combined patient and microbiome metabolites were analyzed from serum and different biofluids, and then antiviral antibodies were also analyzed. So different omics analysis starting from genomics, transcriptomics, proteomics and metabolomics were taken forward to analyze and integrate those data. So you can see that by integrating these different sets of data helps in getting the information related that how it is changing the state, disease state, and can help us in getting the early marker that okay, so microbiome is changing, so patient might be developing diabetes within few days, or to how to administer or how to prevent those, or and also to cure those. So the different viral and microbiome data, genome, transcriptome, proteome were analyzed individually and a combined fashion to determine the dynamic pathways that change during viral infection. So if a virus getting infected, what are the dynamic pathways that changed? In addition, correlation of this microbiome data and those of the patients to obtain a global microbiome host changes that occur during viral infection. So how the global microbiome as well as host changes. So whenever there is a viral infection, so how the normal flora, what normal microbiome level, what is there, so how they are altering with the host level. So this was the study generated. So this is the paper which was published in Cell, that personal omics profiling reveals dynamic molecular and medical phenotypes by Dr. Michael Snyder and his team had performed this. So the sample types were taken as PBMC, peripheral blood mononuclear cells, and the serum. The method was whole genome sequencing, whole transcriptome sequencing, mRNA and miRNA, proteome profiling from PBMCs. So whole genome sequencing, their variant calling or facing, variant expression, RNA editing. So you know that the different splice junctions of the novel isoforms can be found out through RNA sequencing, where different splice altered splice junctions or novel isoforms or the fusions which lead to novel protein products, or the can be done with the RNA sequencing. And if you find out okay that from the proteomics data you can confirm that okay, this is the novel exon or splice junction sites which have occurred, and you can also not only predict but confirm when you do perform an integrated proteogenomics approach. And then the proteome profiling and then variant confirmation in RNA and protein can be done through a comprehensive proteome profiling. And then also with the proteome experiment we can do an quantitative differential expression dynamics to identify what are the different proteins that are getting altered. And from serum what has been done is the untargeted proteome profiling, to identify the quantitation, differential expression and dynamics, and targeted proteome profiling. So after you get the different proteins that which are differentially regulated, or the different cytokines or the different inflammatory agents, so you can directly quantify those using targeted approach. In the metabolism profiling, to identify that how a compound, enzyme, and then finally all the pathways, the compounds which are the byproducts of those reaction, or the molecular pathways or sub pathways, that how they are changing. And then autoantibody profiling to identify that differential reactivity. So we know that our body gives rise to immune responses only when to some foreign particles or some foreign agents, but when in disease, but in certain cases, in diseased conditions, when the person develops antibodies against those particular antigens, or these antigens of the proteins on of own body started behaving as antigens, or autoantigens. So by autoantibody profiling, you can perform the differential reactivity of the bodies in different disease conditions. And also, to serum has been used for different medical lab tests to identify the different biochemical parameters that are regularly being used for the glucose and CRP and everything. So, and finally, to integrate the metabolome, autoantibody, targeted proteome, untargeted proteome profiling, whole transcriptome, whole genome sequencing, to find out the integrated personal omics. So this is the full proteogenomics. This is a full study design for the IPOP, integrated personal omics profiling, which initiative was taken by Dr. Snyder in Stanford, comprised of to identify those fully papers. So if you want, you can just go and read through this. Okay, so here the full article. So how whole genome transcriptome proteome metabolome clinical tests could give insights, RNA edits, SNVs, SB deletions. What are the disease associated weird variants, high interest drug related variants. So you can see the different clustering heat maps are provided. So the dynamic data analysis framework. So if you go and search for the data, so raw data types, data pre-processing, common framework data classification, clustering of enrichment analysis. So differential RNA-seq, differential protein, differential metabolites. Now these three needs to be analyzed in their own way for the QC, for the identification of proteins, for the identification of metabolites, then annotation, then QC, then missing value imputation for the protein and the metabolites. Then here differential RNA-seq will be of PMRF filtering plus quantile normalization, and then you will get to find out, and then you will to integrate those data and try to make sense out of it. And then clustering analysis, then time associated. So all these things have been performed. So this was a full paper which was published to identify this personal omics profiling, and this is one of the first initiative taken for the integrated personalized omics profiling to identify the disruptions in the cells in the disease conditions. So now another was the precision therapeutics in myeloma. So you can see that one was for the cardiovascular diseases and one was for the diabetes. So now where we stand with precision therapeutics in myeloma. So what are these prosperity, what are its promises, and what is are very much still in a stage where we can say that it is a kind of dreaming, or we are on the path but we need to achieve those. So the thing is that, so the different, it has been seen through this omics profiling and technologies that the different patients, if you find out that the different patients can give can have a single can have two or more pathways which can be unique pathways, or which can be common pathways, along with some unique pathways gets altered in certain patients. So for example, if a patient is having RAS and MAPK pathway altered, as well as including a PI3K KTM pathway, and if the drugs are attacking only PI3K pathway, which is general cause of cancer for most of the patients, then that patient which have been given only for PI3K KTM but along with the RAS MAP is also altered for that particular patient, so the patient would not respond to the drug, and from the first picture or the image you could understand that it will happen that only it will show no effect. So among the three graphs where it is in effect or no effect or the adverse effect, so the patient will have no effect, and so but the thing will be like that it is not getting anything is happening by administering the drug which is giving so much positive result for other patients. The reason is that there has been some other alterations also. So if both a combinatorial treatment or targeting both those pathways can be given, then the patient can be relieved or can get cured in a very much better way. So these are the types of pathways, targeted pathways, and the drugs have been taken forward for treatment of myeloma and for the different clinical trials that are going to treat myeloma. So these all pathways have been identified through different omics analysis, and different omics analysis has only helped in identifying the different molecular pathways of the networks that are actually involved in those aggravation of the diseases or the cause of the diseases. So we can go through it where we stand with it's a recent paper of 2022 only. You will get to know about all the recent activities that have occurred, as well as the different markers and the different studies that have been done to identify the markers associated with multiple myeloma. Okay, so now moving on. So integrated omics tools, advances, and future approaches. So we have known and we have seen that the different integration of the omics data can reveal us many features and tell us about many diseases, can help in targeting drugs for different diseases. So this is an article which will give you a glimpse of all the different applications that are present, which helps in analyzing those data, in understanding, interpreting those data uh, to for that which we can report or which we can understand the cause of the various diseases. So okay, so the with the if we just reading the first line, you can understand that what is the importance of these tools and why it is needed. is that with the rapid adoption of high throughput omic approaches, so different high throughput approaches generates a lot of data, but that needs to be curated, annotated, analyzed, and then only it can be interpreted. With the data we cannot interpret, so you have understood that how genomics data is generated, but you need to go through a steps of like QC correction, then identifying, then alignment, then only can interpret the variant. Similarly, the proteomics and the metabolomics or other omics data also requires a set of pipelines or the set of tools through which early can analyze those data. So such as genomics, transcriptomics, proteomics and metabolomics, each analysis can generate tera to petabyte size data files on a daily basis. These data file sizes together with differences in nomenclature among these data types. So these data types are firstly huge. So what you call as big data, or so these data file sizes are huge. Then you need to integrate, and these data type data formats are different for all those three. So these are so integrated omics, multiomics, polyomics, transomics, panomics. So this challenge include differences in data cleaning. So you need this is a difference in into data cleaning, then sharing, then data archiving, then data storing, then data managing. So these are very much, there are very various hurdles at each point of data after your data is generated. So you need to store those, you need to analyze those, you need to curate those, you need to archive those. So ultimate goal is towards the holistic realization of systems biology. So this is comprises of the whole of the systems biology, from where the gene, the protein, the metabolome, finally the gene expression, gene regulation, everything comes under of it. So now commonly used approaches are the integration, interpretation, and insight. So once you integrate those data sets, you will be interpreting those data sets, and once you interpret those data sets, you will give insights into the new diseases, into the new therapeutic approaches, or the new therapeutic models, or the new pathways for a particular diseases. So post integration, these very large data sets entail unprecedented views of cellular systems at exquisite resolution. So the thing that we can understand, the resolution can go to at a very low level, from the DNA at the genomic level, the variants, the gene expression, finally to the proteome and the metabolome level, the compounds which we see, which is the effect of all those things that are happening in the subcellular level through various computational and informatics frameworks, with the continued reduction in costs and processing time for sample analysis. So sample analysis cost and processing time has become very less. So has generated different types of omics data sets are now getting generated. What that are glycomics, lipidomics, microbiomics, and phenomics. So there are, it is getting increased day by day. So this can be led to the integration of these huge kind of data sets is very much useful to understand. So there are unique challenges to specific omics platforms. So all the specific omics platform have their own challenges when you try to analyze those data. So unique challenges, one is from the genomics data, you need to link the genotype to phenotype. So whatever changes are giving critically of the nucleic acid amplification, followed by reliable quantification and molecular annotation based on sequence identity. Current sample provide a means to analyze all our DNA, RNA in a biological samples, all coding and non-coding RNAs. So now the major limitation is the interpretation of in context of biological function, that is the influence of specific variance or phenotypic variation. So you can sequence all the data with the ease of the and the high throughput sequencing technologies. You can sequence all kinds of data in the genomic DNA and RNA level, but how will you identify that what is the effect or what is the extent of the effect of those variants at the phenotype level? So there comes the unique challenge. So combining those data from proteomics and metabolomics will help to understand that okay, so these variants has given rise to this malfunction protein or this altered protein. That's why this pathway or this is not behaving the same as it would have been done in a healthy case or a normal case. So the extent of variance, so you can identify those variants with the genomic sequences, but you cannot understand the extent of this variance until and unless you do a comprehensive proteome and metabolome profile to understand that how this variance is affected at the phenotypic level. Then the quantification of the proteome. So now the proteomics data often provides information, but now you need to quantify this. So there, the instrumentation challenge lies in that if the protein expressed is in a very small volume, you cannot quantify those. You can hardly detect those, but you cannot quantify those. So you need to identify significance of amount of sample. So if you sample is less, you cannot identify the proteins because there is no method of amplification like DNA. With a small amount of DNA you can amplify, but with the proteomics technologies there is proteomics protein samples, there is not so much possibilities of uh, amplifying it. Then the difficulties in isolation of the membrane proteins, detection of low abundance proteins and the insoluble proteins, or the proteins which you cannot digest with the chemotrypsin which are available. So that those all are the challenges of identifying and quantifying proteome. So nuclear proteomics in a proteomics requires enrichment of nuclei. Thus, even untargeted proteomic approaches will not include data for all proteins with a given biological samples. The reliance on separation of complex chemistry, this is different charge states, post-translational modifications using chromatography adds to variability in quantification in top-down and bottom-up proteomics. In addition, there is variability in peptide identification due to variation in peptide structures, charge, hydrophobicity, and these biochemical properties of peptides and proteins affect their and identified by NMR or MS. So analysis pipeline of proteomic data must deal with absent data. So when all the proteins, when you try to analyze your data, there will be some missing values which occur. So due to because of it is not ionized efficiently or it is truly not present in the sample, that you cannot know, that's where you have need to trade off between that. Okay, if my data consists of considerable amount of data in the samples, so then other samples which are missing can be imputed, otherwise you cannot take forward that data and you don't know that it is the fault of the site is sample issue, or that it is not present biologically in the sample, or due to the instrument could not detect it. So what is the case? Then you have to normalize, then you need to find out quantification. So all these things are there. Now there has been effective labeling tool. So when you need to perform a labeling tool or the labeling experiment, so you will definitely use different labeling approaches like ITRAC, TMT or SILAC. But LFQ is a label-free approach where you don't need to use the label techniques and you try to identify label without those ions, and you try to find out the relative quantification of those peptides and then the proteins that gets detected in your mass spec. So this is how different protein quantification can be done. Next is the quantification of the metabolome. So metabolomics data now can further, metabolomics is the ultimate change in the compounds that can be linked the genetic and the proteomic variation to the functional variation and provide novel insights into the metabolomic regulatory and signaling activities which can occur in a given cell or a tissue. However, the similar to proteins, metabolites are not amplifiable and only 15 to 30%. So you just note it down the percentage that you can see that only 15 to 30% of the entire mass spectra are identifiable and quantifiable, the limiting the useful amount of the information generated. So it hence it limits the amount of information generated. In addition, false positives also a challenge. So since the annotation is done through on a probability or a predictive basis, so there are chances of false positives as also. So these are the major challenges. So different lack of standards for data formats and analysis pipelines are major challenges. So large scale efforts in the metabolomics research community are going to challenge including standardization annotation of metabolites and methods and statistical considerations. So workflow in integrated omics is the genotype is the genomics, transcriptomics, proteomics, metabolomics. So you will the effect of genomics to transcriptomics and proteomics to the metabolomics. So then we can have different statistical approaches, and there are clinical and imaging data sets as well as antibody based approaches data sets. And here you will get fluxomics, ionomics, microbiomics, lycomics. So statistical approaches. Now the integrated omics, multi-omics or the transomics. So you will get the novel networks, novel pathways, then models, predictions, then you will can identify those biomarkers. And then you can prediction and of the health and disease, and finally you can predict that which markers, which proteomic or the metabolomic markers can lead to a particular disease, and then you can get the phenotype. So just look at this paper, the different tools that are available till date. So this is the paper. So if you go and search here, I think we should give it in the chat box. Here the tools that are differently available. So here you can see the omics data integration tools like MapMan. So what is the computational platform, what is their friendliness, what is the functional, and its availability. So you can search, and here you can see that it was visualize and map gene expression, metabolite or other data. So large data sets into diagrams of metabolic pathways. Okay, so here it will help you so you can download and you can use these annotations to use different kinds of data visualization tools. Then weighted gene co-expression network analysis or eye clusters, or detection of the novel biomarkers, annotation with existing knowledge is using corresponding transcriptomics and proteomics data sets, or Intomics or the Impala, which you can join pathway analysis of transcriptomics or proteomics and metabolomics. So it will perform over representation or enrichment analysis. 3-omics, multi-omics data matcher ingenuity pathway analysis from QIAGEN, which you need a license for integration and mapping of genomics, transcriptomics, proteomics and metabolomics datasets. Then Galaxy P and Galaxy M for a complete suite for integrated omics analysis. So we had checked that how Galaxy tool can be used for this kind of integrated analysis. Mixomics is an R package which is very much useful for data exploration, integration, visualization of the biological data. So there are many tools. You can try to find out that which tools can be used for your data set to understand and analysis. So there are different resources, and the databases and resources are very much important, where you can get the different genomics data like NCBI and the geo datasets, or array express for the micro datasets. But for proteomics and metabolomics, or proteome exchange or metabolites or metabolomics workbench, which are there for different identification of those different experiments. Okay, so this is for the all of the tools and what are present. So now there is a challenges. So what are the challenges in different integrated omics technologies? One is for the individual omics data sets. So individual omics data set comes with its own kind of data with different phenotypes, different data formats, different data preprocessing techniques, data experimentation setups. So just to buy different scales. So there are different values of expression of the different omics data sets are different, so we cannot just could simply integrate those data sets without having proper scaled or proper annotating or proper identifying. So we cannot just go and scale data sets. It needs a proper rapid scaling and identification of those data sets. So individual omics data sets, normalization, transformation, then the imputation strategies. For example, proteomics and metabolomics data will have contains missing values and missing data. So you need to have some imputation strategies, whereas the genomics data doesn't contain such strategies, such missing values. So we need to understand the data, then the data pre-processing for the amplification of the library check or the quality check for genomics data are quite different than from the proteomics or the metabolomics data. Secondly, yeah, the amount with which we can sequence the genomics data is much less, but for the proteomics we need a good amount of sample, as these are not amplifiable. Then the different instrument to instrument variations, the repeatability, the reproducibility, the stability of the peptide. So when you try to identify the proteins, so the how much the peptides are stable, as well as the metabolites, how much they are stable and how they much will be detected and quantified, it's all needed. Then the experimental challenges, the sample preparation, in the study design, then the reproducibility, steady state, statistical power, that how many samples are there. So if you have sample size plays a huge role in specifically designing this kind of experiments. Where the sample size is less, then the power of your experiment becomes much lesser, and you need a proper validation of the markers that you will be reporting in both proteomics or metabolomics sets. Then is the integration, then the data integration issues, so data scaling and reduction statistical tools. So there are huge chances of getting false positive results, because all these omics technologies generally are on the prediction based. For example, that if you go for the genomics data set, if you remember that it need a QC score of greater than 30, because the chances of the base that has been called is not by chance or random, or it's actually the same base, depends, and from there depends the quality of the bases. Then the workflow, the pipelines on different data sets are through different pipelines, and then and visualizing the pathways and the networks. And after you perform all those data pre-processing steps, try to understand what are the pathways from each data set, and try to integrate with the help of these tools, the different tools that I showed, that different transcriptomics, genomics, or the proteomics, or the metabolomics data sets, and then try to identify those data sets. You need to now interpret those, that the compound, enzyme, then the reaction, the gene responsible for a particular expression level of the protein for the particular expression of the compound, and the compound for the and through the different enzymatic pathways that are having. So interpretation of those results, then to find without then to nullify the effect of the false positives, and determine the biomarkers that are that you can differentiate or classify or cluster in between the disease and the normal conditions. Then validation of those targets, because the chances of false discovery are huge. You need a proper validation of those targets, and then those database for usability or the phenotype prediction. So you need this kind of things in a very focused manner. So all these are very much challenges in performing integrated omics kind of technologies, where you need to keep in mind the experimental challenges, number of samples, how many samples are initially required to satisfy your data, or to have a proper power test, statistical power test for individual omics data sets. What are the data issues? What are the integration issues? What are the knowledge level issues? So it might happen that after you integrate and analyze all those data, it cannot interpret those knowledge. So if you cannot interpret, it won't be of much help to the community or the society. And finally, after getting all this, we can have move way forward for the personalized medicine, where high throughput technologies can be done, the profiling of a patient can be done, and medicine and the death pathways which are affecting for that particular patient can be identified, and the medicine can be provided, so that the so moving back to the first slide, so that our goal of that this effect of the graph of this effect goes on, and this every patient can get a proper cure, treatment, or management of the diseases. So this was for all for the week 9's proteogenomics approach, the personalized medicine. So thank you all. So we will be meeting next on week 10 of the session. So thank you all.